Hello, everyone. We were just waiting a couple of minutes for the attendees to join today's session.
Okay, well, yes, we'll get started with the introduction and opening as more attendees keep joining us. So on behalf of ISB and the organization committee, I'd like to welcome you all to our second session of the 2021 virtual Biocuration conference. I'd like to thank everyone who joined the first session and made that such a great experience for us. We had over 130 people at that session. We're currently up to about 70-ish. And so we'll keep, I think that number will keep climbing as this people joins, but welcome all. Thank you for joining us. My name is Sue Bello, and I am a curator at Mouse Genome Informatics and the Alliance of Genome Resources, and one of the organizers of this conference. And before we get into the details of today's session, I'd like to just thank everyone who volunteered to help us with this effort. And a special thank you to Federica Quegla. I knew I was going to mess your name up. It's early. And her PI for graciously offering us the Zoom webinar that we are using to host these meetings and allow everybody to attend. We plan to have 2 more virtual sessions this year. And we can't hold these without your participation. So please continue to submit abstracts. We've had a few questions. Our intention has always been to roll over abstracts as long as the submitter is willing to have them considered for the next session. At the next session will be in August. And at that time, we will probably have another 2-hour session, but just with talks, rather than the talk panel format we've been using, so that we can actually get a few more talks in. But to do that, we need you all to submit abstracts. And then we will have a poster session in the October session, along with the ISB general meeting and a panel on the past, present, and future of the society. And we're planning to use a different app for the poster session, something that's called GatherTown, which will actually hopefully allow us to have a more in-person like poster experience where you can actually walk up and interact with the poster presenters and look at things and choose to what you want to see and interact with. So please stay tuned for those developments. Also, it's been decided that in 2022, the ISB will not be holding an in-person meeting, and there is a plan to send out a survey about what form the 2022 meeting will take. And so please do give us feedback on that so that we can present an excellent meeting in 2022 as well. So for today's session, we're going to do something very similar to the first session. In the first hour, we will have 4 talks by speakers whose abstracts were submitted and chosen. The talks will run about 12 minutes each, with then 3 minutes for questions. The audience is able to submit questions using the Q&A, or you can submit them in the ISB Slack. There is a Biocuration2021 session 2 channel where you can submit questions, and we'll have somebody monitoring that to feed them to the speakers. And if you're not already a member of the ISB Slack, I will post a link shortly so that you can join. And we invite you to use that to come in. And then in the second hour, we will have a panel discussion on careers in biocuration that is moderated by Peter Uetz. And again, you can post questions for that session in the Q&A or in Slack. So our first speaker will be Jiyu Chen, and he will be speaking on automatic consistency assurance for literature-based gene ontology annotations. And I will hand it over to you.
Yes, thank you, panelists. Thank you for inviting me to give this presentation. So today I want to share with you our recent studies on the topic of quality assurance for gerontology annotation. So our manuscript is accessible on the BioArchive and is currently under the review of a journal. So recently, we have identified challenges in assuring the consistency for literature-based Go annotations stored within biological databases. By integrating curation guidelines and reviewing qualitative reports, we formalize 4 major types of inconsistencies that are currently confusing either human curators or computers. We designed a set of heuristic rules to manipulate a manually curated Go annotation to simulate a certain type of inconsistency and generate a synthetic dataset. We proposed a framework for supporting the development of automatic inconsistency detection model. And this framework is suitable for both a human-in-the-loop curation scenario and can support both rule-based and machine learning-based implementation. And our approach is adaptive to the open world knowledge, such as can address consistency assurance with respect to the constantly updating Go vocabulary and newly published studies. We also integrated a state-of-the-art text mining model and evaluate its effectiveness in detecting different kinds of Go annotation inconsistencies using a set of quantitative evaluation matrix. So a Go annotation can be supported by either a cited publication or a genomic sequence. We typically focus on a specific type of the annotation called literature-based Go annotation. This kind of annotations are produced by manual reviewing of experiment details within publications and are labeled with experiment type evidence code. In most cases, literature-based Go annotations are considered as the most reliable resources in the databases. However, the Go annotation is not a one-time process. Once a gene function information is identified and structurally represented using the Go term, curators need to constantly inspect its quality, such as assuring the gene function information represented by the Go term is consistent with the naturally written evidence findings within the cited literature, or is kept updated with the new knowledge. A recent publication from the Go Consortium database has also amplified that the modern curation work for gerontology is shifting from enriching or producing new annotations towards quality control for existing Go annotations. Unfortunately, it is quite challenging in assuring the Go annotation consistency. Firstly, the reliable manual assurance is inefficient. At present, there are almost 8 million annotations across almost 5,000 species recorded across different databases, but less than 2% are manually curated. It is infeasible for manual curation to keep up with the daily updating studies. And secondly, few automatic tools are presenting strong reliability to increase this efficiency. Most of the strong performing tools can be biased towards frequently selected Go terms or annotated genes. Most of the tools cannot adapt to the evolving studies and updating Go vocabulary. For example, the Go categorizer cannot justify a Go annotation if the Go term was newly added into the vocabulary without having attachment to any piece of text yet. The existing studies were mostly focusing on Go annotation enrichment rather than maintenance. Tools for Go annotation enrichment are not directly applicable to assist in a Go consistency assurance because identifying low quality annotations is more difficult than finding a relevant Go term for producing an annotation. For example, the dictionary-based method can recommend a Go term as a candidate to curators for producing a Go annotation if it is mentioned as a keyword within a short piece of text. However, the occurrence of Go term do not imply it is a truly valid Go annotation. Intuitively, We notice that many Go annotations with inconsistent representation of the gene function information is very close to a consistent or correct representation in terms of confusing human and computers at the same time. So the existing methods do not show potential in distinguishing annotation between consistency and relevancy. We will also give examples of such confusing Go annotations in later slides. The third challenge is that there are lack of qualitative or quantitative studies, especially on analyzing the scale, the scope, or the characteristic of inconsistent literature-based Go annotation records within the databases. Although many papers were discussing various aspects of the quality concerns of Go annotation at a very high level. So we will introduce 4 major types of Go annotation inconsistencies, intuitively inferred by reviewing different Go annotation guidelines and curation handbook. These 4 types of inconsistency were caught by us because they are confusing either human curators or challenging the automatic methods. The blue texts are a naturally written text directly extracted from a full text article stored within the PubMed. The red text is a Go term being selected to represent the gene function information where the blue text is describing. The first type of inconsistency is confusing computers at most. The evidence in the cited publication is introducing a positive regulation of vesicle fusion, while the selected Go term for annotation is describing a negative regulation, which is contradictory to the evidence. The second type of inconsistency is confusing both human and computers. It is inferred by a key Go curation guideline that curators need to select a Go term as specific as possible to describe gene function information, but shall not overtelling the information. The provided evidence in the cited publication do not support that the gene function is an endosomal vesicle fusion, while the selected Go term for the annotation has overrepresented this information. Thus, this is also considered as a over-specific Go annotation. The third type of inconsistency is mostly confusing computers as well. We notice that the evidence text in the cited publication has a mention of a Go concept called cytosol, but does not actually express a valid cellular component information of gene product. Thus, we consider that these evidence texts do not correctly support the gene information described by the Go term. The last type of inconsistency is confusing to both human and computers as well. The evidence text indicates that the studied gene product is an allelic variation, and the experiment is conducted by comparing the single gene to the alleles of the same gene. Thus, a better evidence code shall be inferred from mutant phenotype instead of inferred from a genetic interaction. We call the previous 3 types of inconsistencies as term inconsistency because it happens between the Go term and the evidence text. We call the last type of inconsistency as code inconsistency because it happens between the evidence code and the evidence text. Next, we will introduce the implementation of automatic methods to distinguish consistent and 4 different kinds of inconsistent Go annotations. Firstly, we will introduce how the dataset is created. It is very time-consuming to manually identify and collect inconsistent annotations from the database. To overcome this challenge, we designed a set of heuristic rules to modify manually curated Go annotation corpus that was initially created for the Biocuration 4 task, and to simulate a certain type of inconsistency introduced above. We use a deep learning language models to take a Go annotation as input and output whether it is a consistent annotation or belong to a certain type of inconsistency. We also use standard quantitative metrics to evaluate our model's performance and proposed 2 strategies to study the suitability of our methods to be applied in a human-in-the-loop curation scenario. So the left side demonstrates a manually curated Go annotation sampled from the corpus created for Biocuration4. Each annotation contains a gene product information, an evidence text directly extracted from the full text article, a selected Go term, and an evidence code. To simulate type A inconsistencies, we scan through all the consistent Go annotations within this corpus and swap the mention of any positive regulation with negative regulation and vice versa. For example, given the consistent Go annotation shown here, we will swap the Go term above with the Go term below. We use 2 steps to simulate type B
2-minute warning.
Yes. Inconsistent. Firstly, we retrieve all direct descendants with either is-a or part of relationships of the Go term in a consistent Go annotation. Then we randomly select one of the descendants and ensure it is not annotated to the same gene product supported by the same piece of evidence text. The selected descendant Go term will replace the original Go term and simulate over specific inconsistency. We also use 2 steps to simulate type C inconsistencies. Firstly, we apply a concept mapper to identify the Go terms in a full text of a cited publication. Then we replace this evidence sentence in that consistent Go annotation with another sentence in the same article. This sentence is containing the mention of the same Go term in the consistent Go annotation, but is not labeled as a valid supportive evidence by the curators, and it will simulate an unsupportive inconsistency. And finally, for type D inconsistencies, we just randomly replace the original evidence code with another code. On the right side, we show the statistics of the created data set. More importantly is that we ensure that the articles for creating the training set and development set is independent from the test set. And we also confirm that a large proportion of the Go terms in the test set do not occur within the training or the development set, which can simulate an update in the Go vocabulary. We model the inconsistency detection with a multi-type single class classifier. So this relies on 2 independent assumptions shown here. At current stage, we assume that the described gene product is consistent between the Go term and evidence. So we only focus on detecting the contextual inconsistencies between the evidence in the cited publication and the Go term. We use a text mining model with deep learning architecture to do the task. The input of the model will be the raw text of a standard Go annotation, which consists of the evidence text, the concatenation of the Go term and Go definition, or the evidence code. The output will be a single consistency or inconsistency class. Apart from the standard quantitative metrics for evaluating the model's performance, we typically introduce 2 quantitative metrics for evaluating the model's effectiveness to a real-world human-in-the-loop curation scenario. We typically focus on 2 aspects. One is we believe that a model is more effective if it can perform better on more certain predictions, as human curators can pay more attention to uncertain output of the model in the real application setting. And the uncertainty is quantified using the Shannon entropy. For the second aspect, we notice that some shallow linguistic features can significantly influence the model's performance. So we summarize these special features into a uniform linguistic test suite and quantitatively evaluate their impact to the model's performance. A detailed empirical analysis can be found in the paper. To save the time, I will only summarize the key findings of our experiment. So in terms of the performance, the model is competitive in detecting term inconsistencies. It is also effective in assuring the annotation inconsistency with open world knowledge, such as the update in the Go vocabulary and visible for human-in-the-loop curation. However, the model is not very promising at detecting the code inconsistency. And because of the bottlenecks of the existing language models, it is less reliable if the valid and invalid Go terms are highly semantically similar or related to each other. For example, such as distinguishing between a feeding behavior and drinking behavior to a piece of evidence text. In future, we plan to extend our study on a broader biological perspective and improving the modeling performance, such as covering more species and more types of inconsistencies, because the original corpus that we're currently using are quite narrowed. In most of the cases, PubMed only provide access to the abstract instead of the full text articles. So we also want to study whether the annotation inconsistency can be detected using the abstract evidence alone. We also wish to make cooperations with database curators and address some qualitative evaluations in future. This work
we are definitely at time here.
Yeah.
You do have a couple questions. I will leave the acknowledgements up there. It's very interesting work, and you.
We do have some questions that we want to ask with the communities, and I am demonstrating that here. So yeah, so that's all my presentation for.
Okay, so we have maybe 30 seconds left. There's a question from Charles Tapley-Hoyt, who is asking, they've encountered type C annotation issues in all sorts of curation relationships. And what are we to do if the mention of the gene is in the introduction or somewhere else in the text, but it's obvious from that all of the text in the article is about that gene? It seems to happen quite a bit.
Oh, yes. Yeah, thank you. That would be very helpful feedback.
And I'm going to let you answer the other questions in the chat, if you wouldn't mind, so that we can go on to the next speaker.
Yes. So
thank you very much. That was a lovely talk, and we really appreciate you getting up in the middle of the night to come join us here live.
Yes. Yeah, thank you.
So our next speaker is Sushma Naithani, who is going to talk about biocuration training for undergraduate students, challenges and opportunities. And Sushma, if you'd like to unmute yourself and share your screen.
Hello, good morning to everyone. And I'm going to share my screen soon. So can I? All right. Can you see my PowerPoint presentation?
Yep, your PowerPoint looks great.
All right. So hello, this is morning here in Oregon. Greetings to you all from Corvallis. And today I'm going to talk to you about the biocuration training for undergraduate students. So before I talk about challenges and opportunities, I would like to share some of the work that was done by undergraduate biocurators in my lab. I started working with undergraduate biocurators in 2012. My first two students, they both worked with me about 2 years each. Eli Weddle, he was a biocurator who worked with Uetz, and Christina Partipello worked with Fragaria site. Uetz and Fragaria site, they are species-specific metabolic pathway networks. And each of these students helped to edit, biocurate about 100 metabolically important pathways for each of these species. If you have interest in using any of these metabolic pathway databases, they are available through our website. Link is here. And more information is also available in the 2 papers that we published with undergraduate researchers, as you can see here. So I also worked with another student, and he did some biocuration work with the gene family. I think especially the manual biocuration approach is more appropriate for biocuration of gene families, and I'll tell you why. So typically plants, I work with plants. Plants have a lot of gene duplications and polyploidy events. So we have large gene families in plants, and not all the genes are well characterized. So typically what happens, we have 1 or 2 prototypic member of each gene families, and whatever their function is or their Go annotation is, that is propagated to most of the other family members. So we looked into one of the subfamily of receptor kinase that is known as S-domain kinase subfamily. It has about 144 genes in rice, and most of them have Go annotation related to the prototypic member of this family, that is S-locus receptor kinase or SRK. It was first characterized in brassicaceae, and it works as a female determinant of self-incompatibility response in brassica. Now the SI is very limited. This type of self-incompatibility is very limited to crucifer family, and rice is a self-pollinating crop, so SI doesn't exist here. And so what is happening to this 144 genes in rice? Are they redundant? Do they have any functions? So we did literature review, and we also analyzed publicly available omics data, and we assigned more functional annotation to these genes and proposed a new or improved nomenclature for these 144 genes. So this was the work done for honor thesis by Raymond Dykeman. Then I worked with many students on genes and pathway curation on plant reactome. So last year I had 2 students who each worked on pathway biocuration. However, after a few visits in the lab, we had the pandemic, and most of the work was then done through online collaborations and Zooms. So that was a new experience for me. They worked from February, then from March to June, we worked only through online. And each of them were able to curate 1 or 2 metabolic pathways for plant reactome. This year, we were prepared to work online a little bit more, and I had 6 students who worked with me. So one of the students who had more background in coding and was interested in learning biology, so he worked on the project. Especially, he looked into the data that we have behind the plant reactome. So little bit about plant reactome. So plant reactome is a plant pathway database of Grameen project. We use rice as a reference species for curation, and then we use gene orthology based projection to another 106 species. We have done this curation now for about a decade, and he wrote a lot of his scripts that were used for quality control and checking the consistency of the biocuration. And also it gave us opportunity to look into evolutionary history of various reactions and metabolic pathways across all these 106 species. And then I had the 5 students who were all set to do biocuration online. And so we kind of had a virtual classroom for doing biocuration together. And we did biocuration of genes and gene networks involved in various abiotic stress response. So now the challenge this year was not standardizing the data and curation practice, but how do we set up an online virtual hands-on classroom for doing biocuration? So we had some tools and some experience in our pocket to start that. And I'll tell briefly about that. So a little bit about our approach. So our approach to biocuration is knowledge-centric, which means that we spend a lot of time in critical review of literature, talking about the various scientific methods and evaluating evidence, synthesizing knowledge from different sources, and gathering data about genes, how and where genes are expressed, subcellular locations of gene products, whether they are RNA or whether they are proteins, which reactions they are associated with, and can we associate 1 or more reactions to make a pathway, and then summary of the genes, reactions, and pathways. We also like to link the complementary information that is available in other databases and refer them. And if we improve on, for example, any gene annotations, then we submit to those respective databases, for example, for rice, RepDB. So then students also spend some time navigating at least the RiceDB, Grameen, UniProt, TrocPal, Plant Gene Expression Atlas, of course, for literature, PubMed, and then PlantMap to learn about various genes and proteins and how and where they are expressed. So our first 6 weeks actually go deeply into discussing literature, reading together, and navigating different information available in these various resources. So some of the strategy and training material that we use for this training, in addition to that, comes from our previous experience of conducting pathway curation jamborees in 2017 and 2018. Also comes from my teaching undergraduate students and working with them for about past 20 years. So it helps me to articulate learning outcomes for students. And then we use some of the training material that is available from EMBL, EBI, from Plant Reactome, from Grameen and UniProt. So I like to share this list with you. If you are a biocurator and trying to work with undergraduate students, these are good resources. So after reading and collecting, then we start gathering information on Excel sheet. Once it is done, then we enter in our tool. So we use the Reactome Curator tool for doing pathway curation. So now we focus on the tool after we have focused and gathered the knowledge. We start building reactions and pathways. So this is our biocuration step by step. So I like to summarize my experience working with undergraduate students. So of course, there are challenges with working with undergraduate students, and these challenges, they have no prior experience. They lack fundamental scientific concept and vocabulary. They have also limited time. So some students will only work 5 hours, which means maybe 2 hours or 3 hours twice per week. And then more training and constant supervision is needed. What I learned as an instructor is that teach one concept at a time, assign one task at a time, support exploration, discussion, and reflection, be flexible, but have very clear guidelines and schedule, and then help the student to build their network with their peers and other lab members, and provide opportunity for presenting research work. So here, I like to share some of the biocuration stories that we did in 2021 during the pandemic. These 3 minutes presentation from each of our students are available on Grameen YouTube channel. And here is a link. I like to end this presentation by playing one presentation of a student.
Hello, and welcome to my lightning talk about my project. This year, I participated in the URSA Engage program with my mentor, Dr. Sushma Naithani, and I worked on the biocuration of a rice gene network involved in abiotic drought response. So the first question you might be asking is, what is biocuration? To put it simply, biocuration is using publicly available information from sources such as databases and scientific literature to understand genes and proteins better. As researchers, we synthesize that knowledge from these sources to create a useful representation of the data. By integrating data from these sources into a database, we make it more accessible for other scientists. As you can see in the database diagram to the right, there's a lot of information to curate. This is a pathway diagram of the species I worked on, Oryza sativa. This example is the ideal of biocuration, an information-dense but relatively simple map of the gene interactions in a pathway. The research process of biocuration involves extracting and interpreting data from scientific publications. In my case, I made use of some information from Dr. Naithani's lab that they already had available and added from my own reading of scientific papers. This initial step helps us understand gene relationships and is where most of the lab work comes in. The next step in biocuration is to add in all the other information we find from other databases. One large part of this is ensuring cross-compatibility between databases. Information isn't very useful if people can't agree on what gene we're talking about. Since every database has a different naming scheme, a well-curated gene will have references to all of these names. In addition, many other factors about a gene go into a database entry, such as pathway information, location in a cell, the interactions of the gene, and the actual amino acid sequence of the protein it produces. So once all this information is assembled, where does it go? Once we have an understanding of the gene and the pathways it's a part of, this information goes into the Plant Reactome database. Here's an example of the types of information we collect for each gene before it is entered in the database. I fill out an Excel sheet like this for each of the genes I curated. I'm still in the process of integrating this information into an entry for Grameen.org. These are some of the genes I'm working with and filling out a database on. For each of these genes, I found the subcellular location, ID in various databases, and in some cases, a brief summary of the gene, among other information. This first slide shows a small summary of the upregulated genes by OSNF-YA7, where the ones inside the smaller circle are present in the nucleus and the ones outside are present in other organelles. This slide shows the downregulated genes for OSNF-YA7. Over the next few weeks, I'll be working with Dr. Naithani to integrate this information into the plant reactant database at gramine.org.
So I'm happy to answer if you have a question. I see one question in the chat.
Yes, so thank you very much for that interesting talk, Sushma. Yes, there's one question in the chat, which is asking about whether you have any curation standards or guidelines that you follow that could be applied to any curation task. You know, they've observed, this is from Charlotte, and I've observed most of the time standards change from lab to database to the other, and was wondering if some consensus existed somewhere. And that is a very good question.
Yeah, this is a very good question. And so, you know, at least I think that the problem we face is about the gene IDs, and many organisms have the same problem. So in literature, you find sometimes the gene names, sometimes you have the systematic locus ID, sometimes you have some other nomenclature. And so that is not very consistent. And when you try to synthesize information, one of the first target is that how do you relate A to B to C? And so we spent a lot of time in mapping and building consensus on the gene IDs. So I think that goes across everything. And I think the other part, whether it is human, yeast, plants, is that if you are talking about evidence that this gene is here or located, say, for example, in mitochondria, what is the evidence? Or if it is located in plasma membrane, what is the evidence? What is the really done? When you talk about protein-protein interaction, then also it is very important if it is coming from prediction, if it is coming from yeast to hybrid, if it is coming from isolating a protein complex, and there is a priority why a certain method can be relied more and used as a final statement as compared to the other. So really looking into biology is I think the key. You can't have all the evidence parallelly and assign them the equal weightage. So for example, I looked into SUBA, that is Arabidopsis subcellular localization database. And I was looking for localization. And so they had about 8 or 10 prediction-based cellular localization assignments. And then they also have GFP tag and they have some MSMS data. And then when they say the final winner, they put the experimental evidence as well as the predictive evidence together, and where you have the majority of consensus, is there a subcellular location? And that is not true. So I go into that, and then I look what is the information coming from GFP tagging experiment, use that as number one, and then MSMS information, if that is available.
Okay, well, thank you very much. We are at time. If people have further questions, you can put them in the Q&A or in our Slack, and you can, then we'll try to answer things after the talks. For the panel, for the remaining speakers, I'm going to try to just turn my video back on when you're at the 2-minute mark, when you have 2 minutes left to speak and before the 3 minutes for questions, rather than trying to interrupt your talk. Our next speaker up is Olivia Perfetto, who is going to talk about a resource for the network representation of cell perturbations caused by SARS-CoV-2 infection. And your screen is already shared, so excellent. Take it.
Thank you, Sue. So yes, I'm Livia, and I work in the University of Rome Tor Vergata. So my supervisor is Professor Gianni Cesareni, and I am the senior coordinator. So actually, I work under the collaboration between Human Technopole and the University of Rome Tor Vergata, and we are a small group who hold and maintain and curate different database of molecular interaction, so functional and just protein-protein interaction. So for instance, Ignore and MINT, but also MENTA and others. So, and today, so I want to give a little bit of an introduction. So a little bit about a background about the resource I'm presenting today, for which we have, so we have done this work of curation of causal interaction related to SARS-CoV-2 infection. So the database is Signor. So Signor is manually curated that resource, annotating logic relationships between human entities, mostly proteins. So we annotate logic relationships, meaning that the data structure so that we adopt, so the format type that we adopt is the activity flow, where we have binary interactions. We also, so every interaction is also directional, meaning that we have a regulator and a target, and it's also signed, meaning that we can have an up or a down regulation of the target. So again, it's a manually curated resource, and we annotate interaction which go and compose a large interactome counting 28,000 interaction arising from almost 10,000 publications. And as I will show later, we also annotate metadata and additional information like post-transcriptional modifications. So something about the structure of the data that we host. So every interaction in Signor is composed by three main parts. So the part concerning the entities, so the molecules that form the binary interaction. So we have essentially proteins, but we also have protein complexes, protein families, phenotype, stimuli, chemical compounds, and so on and so forth. Secondly, we have information, we annotate information about the relationship. So basically the minimum is the effect, so the up or the down regulation of the target. But for the vast majority of the interaction, we also annotate metadata, such as the mechanism. For instance, if the upregulation occurs following the phosphorylation of the target. And whenever it's provided, we also annotate the modified residue. So the setting in this case, which is also in this case, which is modified by the kinase AKT1. And thirdly, we annotate the reference, so the literature, the piece of evidence that support the interaction, for which we provide the PubMed identifier plus a short sentence that resumes the interaction and should help the user to confirm the interaction. So in addition, so every interaction in Signor can be displayed as this tabular format, but also as a graph viewer. The graph viewer is organized in different compartments. We have the nucleus, which is this light purple down here. We also have the plasma membrane, and we have the extracellular space. Finally, we have something like the cytoplasm and an hyperspace to annotate phenotypes. So as you can see, we have different types of entities, like these reddish rectangles over here are the phenotypes. These are the stimuli, and we also have the complexes, and these green nodes are proteins. And the annotations on protein entities are automatically placed in these compartments. And some of the information comes from gene ontology annotation, like the extracellular space, the plasma membrane, and others come from resource that annotates transcription factor, which we usually place in the nucleus, but there is no transcription factor in this case. On top of general manual curation of interaction, we also perform pathway annotation. So we annotate pathway, like in this case, autophagy is displayed. But we want to stress that also pathways in reality are a subset of interaction coming from the big interactome. So these interactions are also placed in the big picture of senior interaction. we provide our resource is completely freely available for download, which is available as a very user-friendly tab delimited file for which we have our own control vocabulary for entities and relationships. But we also follow a community-defined standard. So our interaction are curated in accordance to the My2Cast, so the minimum requirement for annotation of causal interaction. And we export our data as causal tab. So a newly recently developed PSIMI tab 2.8 developed to annotate, to represent causal interaction. So these standard has been developed under the umbrella of the PSIMI and the Greek C consortium. So a little bit about the entities. So as I mentioned before, we have different types of entities. So they are growing in number, and we try as much as possible to cross-link to external resources like UniProt or KB, PubChem. We use complex portal AD for complexes, but sometimes we have to use our own internal identifier, especially for phenotypes, stimuli, and protein family. But as much as possible, we also try to cross-link our internal developed entities to gene ontology whenever we can. This is a bit more about the control vocabulary of effect and mechanism. And now let's jump to the very topic of this talk, which is this curation effort that we have undertaken during the lockdown back in April 2020. So I don't need to explain much of about the reason why we did that. So, but mostly let me just tell you what was the criteria that we adopted to undertake this curation campaign. So we know that when a virus infects a cell, it tends to interact with cellular components, proteins, organelles, and it modifies and affects the molecular pathways of the target cells, leading to a certain phenotype change. So following the reasoning, we decided to do two parallel curation. So the first curation approach was to cover the pathways, the cellular pathways that were found to be affected under coronavirus infection. And the second effort was done to try to cover the causal interaction downstream of cellular target of viral protein. So for the first task, so we we create a number of cellular pathways that we call, so inspired by the hallmarks of cancer, we call the hallmarks of SARS-CoV-2 infections. So we curate these 10 cellular pathways that are known to be affected by coronavirus infection. Everything is available in a dedicated page in our website, which is senior.uniroma2.it/COVID. So every box here is a link to a map. So every map is available as a cytoscape map and in our graph viewer and freely available for download. So as you can see, it's a general senior map. So with it's a general pathway where we have complexes and phenotypes and proteins. On top of that, we also annotate the effect of viral proteins on human targets. So the viral proteins are now in yellow. And so we know what are the human targets of these proteins, and we can, in a way, foresee what are the effect of these interactions. On top of that, finally, we also annotate the effect of chemical compounds that have been tested to fight coronavirus SARS-CoV-2 infection. So we have 10 of these pathways, and we also have a global map combining together the 10 of them. Everything is available in the website and also in index. So, and it's completely free to be used and downloaded and manipulated. The second effort was the one of trying to cover causal interaction downstream of cellular target of coronavirus of SARS-CoV-2 proteins. So we started from these three works, the one of Stukalov, Gordon, and Lee. So who did similar work in trying to co-purify using tandem affinity purification, tandem mass spec, human target that binds to viral proteins. So there is a very small overlap between these three works. So there are only 75 proteins that are shared by at least two works. So we decide to try to curate downstream effectors of these 75 proteins. And for 68 of them, we succeed. So the reason we did that is that when we try to did a general pathway enrichment analysis for the 75 proteins, we only obtain a very poor and not very informative enrichment. This occurred because only the 40% of these proteins have an annotation in KEGG and Reactor. So we thought that by integrating this information with the information with the causal interaction in Signor, we could in a way obtain a new algorithm to measure pathway proximity. So basically we developed this new approach to calculate the significant proximity of a list of proteins to a number of pathways. So basically the idea is that we have an input list of genes. We can walk through the causal interaction in Signor to find closer pathway. So summing up, we curate 10 new pathways explaining cells' response to coronavirus infection. We also annotate 1200 interaction downstream of coronavirus targets and everything. So we developed also this new algorithm to measure pathway proximity, which is available in our recent publication. So I'd like to thank all the contributors of this work, especially our developers, Elisa and Priska, and our brilliant curators, Marta and Luana, all Gianni, the leader of the project, and Francesca, and all the students that helped us during the lockdown in doing this work. And all, of course, all our funding agency, and you for the attention. I'm very happy to take any question now.
Thank you very much, Livia. That was a very interesting talk. So we do have a couple questions in the chat and in the Q&A. We have a question from Alex Sticco. I apologize to everybody whose last name I've just butchered this whole time. I keep trying. He's asking, do you use standardized vocabulary for the mechanisms and cell lines? And if so, is it an in-house or is it something public? Or I guess, is it in-house that is public?
Okay, so we use brand IDs for cell lines. So we refer back to this ontology, at least for cell line. And for the mechanism and the effect, so we developed a control vocabulary in the PSI-MI control vocabulary. So there is a new branch for causal interaction, so every term is defined and cross-referenced to the a PubMed ID, has its own identifier and its definition. So perhaps.
Okay, and then Alex has a follow-up, which is speaking more generally. He's curious about what vocabulary needs you've run up against that are not being met by existing resources. For example, why did you find you needed an in-house standard for protein families instead of using families from PDB?
Well, okay, the honest question is that perhaps we were not aware of the families in PDB. But perhaps on top of that, so sometimes we found ourselves in the need of having a more agile way to display things. But of course, we will now on consider to use families from PDB as well.
It is always a challenge to know where everything is. And then we have one more question that's in the chat. which is asking if the data is available in the PSI QUIC, and also are the edges in the network defined and queryable?
Sorry, I didn't get the. Sorry.
Is the Signor data available in the via PSI QUIC? It's in the chat panel.
Yes. Sorry, sorry, Sue. It will be. So we're working together along with the Intact developer. So it was in the plans to develop this new psychic to integrate also causal interactions. So it's a bit difficult to allocate resources in the latest time. So I don't know when it will happen, but hopefully it will be sooner rather than later.
Okay, well, thank you very much for a very interesting talk. I think we need to move on. You definitely have some more questions in the chat. In particular, Alex, there's a number of questions about families from PDB and a desire to have a link to those. But we're going to move on now to our final talk, which is from Sylvain Poux. I probably slaughtered another last name. Functional annotation of specific protein products in the UniProtKB Swiss-Prot.
Thank you.
Take it away.
First, I'd like to thank the organizer to give the opportunity to talk about functional annotation of isoforms and process chains in UniProtKB Swiss-Prot. The mission of UniProt, the Universal Protein Resource, is to provide a scientific community with a comprehensive, high quality and freely accessible resource of protein sequence and functional annotation. UniProt proposed different resources that I will not describe today, and will be focusing on UniProtKB, the reference protein knowledge base, which contains protein sequences associated with functional information. UniProtKB is composed of two sections, UniProtKB Swiss-Prot, that I will name Swiss-Prot for simplicity, which contains biological information reviewed by expert biocurators, and UniProtKB Tremble, which contains automatically annotated protein sequences. What is Swiss-Prot? In the Swiss-Prot section, the biological information is extracted from the scientific literature by expert biocurators or come from curator-evaluated computational analysis. In order to provide an accurate and comprehensive representation of biological knowledge on a given protein, and to make this data accessible to the life science community, we encode biological knowledge in forms that both humans and machines can understand and exploit. What type of information can you find in a Swiss-Prot record? You can find one or several protein sequences, which are products of a given gene, information on the family the protein belongs to, information on the biological function, and if the protein is an enzyme, information on the catalytic activity. Catalytic reactions are in a structured format. We use RIA, a comprehensive expert-curated knowledge base of biochemical reactions that use KB ontology to describe reaction participants. We also have information on the subcell location of a protein and many other aspects that describe a protein, such as post-translational modifications, PTM, involvement in genetic disease, etc. The source of information is provided via evidence tags. And when the evidence is based on experiments, we indicate publications that support the annotation. When the evidence is inferred by similarity, the source entry is specified to indicate from which entry information is derived. In Swiss-Prot, all information presented in an entry has been curated by expert biocurators who are biologists or biochemists. As far as possible, all protein products encoded by one gene are described in a single entry. As you can see here, splice isoform mitochondrial and cytoplasmic from human FH are displayed in the same Swiss-Prot entry. The same is true for functional protein cleavage products. In the example shown here, two distinct protein chains are produced by proteolytic cleavage. The fibrillin 1 chain, which covers most of the protein coding sequence of the FBN1 gene, and the asparagine chain, which is much smaller at the C-terminus. In fact, many publications describe functions that are performed by specific splice isoforms or distinct protein cleavage products. Most human genes are alternatively spliced, and a study on protein-protein interactions showed that alternatively spliced isoforms from the same gene have highly different interactions profile and appear to behave as if encoded by distinct genes. The same is true for protein cleavage products from the same gene, which can have completely different functional characteristics. It is therefore essential to accurately capture the biology of protein products and support the association of functional annotation with individual protein isoforms and chains to provide accurate link between the sequence and the function. To provide this, we recently improved the representation of splice isoforms and protein cleavage products in Swiss-Prot. We adapted the UniProtKB format, text format, and this data is available in all formats, text, XML, or RDF. We provide functional annotation for specific isoform by using the unique isoform identifier, or splice isoform, thereby associating an annotation with an isoform product. And similarly, for cleavage products, we use the unique feature identifier of protein chains, allowing to associate an annotation with specific protein cleavage products. I will now illustrate the importance of individual protein isoforms and chains with some examples. As shown by a human CYP4F3, different isoforms can have different substrate specificity and catalyze different enzymatic reactions. For example, isoform 3A catalyzes predominantly the omega oxidation of oxylipins and displays higher affinity for leukotriene B4, while isoform 3B mainly catalyzes the omega oxidation of polyunsaturated fatty acids. Using the isoform identifier of isoform 3A, we specify its activity in a separate function comment and detail the catalytic reactions mediated by this isoform using RIA. We do the same for isoform 3B using its unique identifier. Second example concerns isoforms that can catalyze a reaction in a different direction, as is illustrated by fumarate hydratase. In this case, isoform mitochondrial catalyzes the hydration of fumarate to L-malate in the Krebs cycle, while the cytoplasmic isoform mediates the opposite direction and catalyzes dehydration of malate to fumarate. We use the mitochondrial isoform identifier of fumarate hydratase to specify that this isoform proceeds the backward reaction and catalyzes the hydration of fumarate to malate. And in contrast, isoform cytoplasmic mediates the forward direction and catalyzes the hydration of malate to fumarate. Or isoform identifiers are used for manual curation of Go terms based on experimental data, thereby providing accurate links between sequence and function. Protein cleavage products from the same gene can have completely different functions, as shown by the human FBN1 gene, which codes for a secreted protein precursor that is cleaved to yield two mature protein chains, fibrillin 1, a structural component of the microfibrils of the extracellular matrix, and asparagine, an orexigenic hormone involved in hepatic glucose release, which is an attractive therapeutic target for type 2 diabetes and metabolic syndrome. Interestingly, fibrillin-1 and asparagine are associated with distinct genetic disease. Defects in fibrillin-1 are associated with disorders of the connective tissues, such as Marfan syndrome, while asparagine is associated with congenital lipodystrophy syndrome. We use the fibrillin chain identifier to specify that this chain is a structural component of the extracellular matrix. And using the asparagine chain identifier, we report that this chain is an orexigenic hormone that targets the liver to increase plasma glucose levels. We also specify that asparagine chain is involved in congenital lipodystrophy syndrome, characterized by a progeroid facial appearance due to the lack of subcutaneous fat. Chain identifiers can also be used for manual curation of Go terms based on experimental data. The most extreme illustration of this principle comes from the viral polyproteins, where a protein precursor is subsequently cleaved into a variety of chains with distinct functional roles and interactions in the viral cycle. And a good example is provided by the replicase polyprotein 1AB of the SARS-CoV-2 virus. This polyprotein 1AB precursor is subsequently cleaved into 15 different chains with completely different roles and localization. In UniProt, we display the different chains in the UniProt entry and describe the function of each chain in a separate annotation fields, such as function or subcellular location. As you can see, our chain identifiers are used for annotation by other resources, such as Intact, Complex Portal, or as well as GoCam in the future. We already created some GoCam models for SARS-CoV-2 in a test phase, and we are now discussing with Go developers to import our chain identifiers in Noctua. To summarize, we improved the representation of individual protein products in Swiss-Prot to facilitate the assignment of functional annotation to the correct protein sequence. These enhance the value of these annotation in downstream applications, including bioinformatic tools that use sequence properties to infer functional annotations. Annotations of specific products is present in around 5,000 Swiss-Prot entries, including more than 1,000 human entries. In human, around 2,000 annotation comments are specific to a splice isoform, and more than 550 comments are specific to a specific protein cleavage product. To finish, I would like to thank the UniProt teams at SIB, EBI, and PIR, and I thank you for your attention.
Thank you very much, Sylvain, for this very interesting talk. We do have a question from Sashi Pujar.
Yeah, I just have to look at.
Yeah.
Oh, okay.
I was going to read it. Yeah, so we'll answer this one live. What are the parameters that decide the canonical isoform designation?
Excellent question. We sometimes have a consensus with other resources like CCDS for human or mouse, but the clear answer is that there is no strict rule, and that we frequently choose the longest isoform as the canonical one. But then we are open to discussion, and we frequently swap the isoform, canonical isoform, when user requests this. And there are sometimes the canonical, and frequently the canonical is the best studied one. But that's true that it's not always 100% the case.
Okay, and we have another question, which is asking, in the case where a feature, e.g. PTM, is detected in a particular splice isoform, does the annotation propagate to all isoforms that contain the site?
No, because the PTM can be annotated on the isoform sequence, which means that only this isoform will have this annotation. But I also have to say that then we need papers that specifically show that this PTM is specific to this isoform. When it's just that they found that this isoform, which is common to many other isoforms, contains, for example, a phosphorylation site, then we might propagate this. But if a paper says this is specific to isoform 2, we'll annotate on the sequence of isoform 2 and not propagate to other isoforms.
Okay, thank you very much for an excellent talk. And I'd like to thank actually all of the speakers for a really excellent session this morning, afternoon, evening, depending on where you are, and thank the audience for all their questions. And then I will hand it over to Peter to begin our panel session.
All right, hello everybody. I hope our panelists can come on online as well. We have a panel discussion about curation careers, or careers in biocuration, with a very illustrious group of 3 panelists. And I think we can use the gallery view. So I'm welcoming Pankaj Jaiswal from the Oregon State University in Oregon, where he's a professor in plant genomics and curating and organizing a couple of resources and databases, which we will talk about probably. Then we have Tanya Berardini, the co-founder and chief scientific officer of Phoenix Bioinformatics in Fremont, California, whose claim to fame is that they managed to develop tools to commercialize public databases, which is really important because a lot of databases do not have much funding, so they have to develop income streams. And then we have Nicola Mulder, a professor at the University of Cape Town in South Africa, who is one of the directors of the Human Heredity and Health in Africa group or foundation, which organizes bioinformatics projects across all of Africa, which is quite a thing. And she's doing lots of other things as well. So what we will do now is we will have short introductions to all our panelists. So the panelists will introduce themselves. And I will start with myself. I'm Peter Uetz, a faculty member at the Center for Biological Data Science at Virginia Commonwealth University. I've worked on protein-protein interactions and protein functions in microbes as well as viruses. And I'm also working on biodiversity databases, especially the reptile database, which is quite a different story. And so I will just pass on to Tanya now, because she's on the top left window of my screen. So Tanya, please introduce yourself.
Right. Good morning, afternoon, evening, everybody. It's lovely to see so many of the faces, well, not the faces, but the names of people that I've worked with over the years and that I've learned so much from. So back up, after a PhD and a postdoc, I joined the team at the Arabidopsis Information Resource as a curator, not really knowing what a curator was. And many years later of serving the plant genome community through that resource, when we went through a funding crisis for people from TARE, and we were down to like 4 people by then, decided to found Phoenix Bioinformatics as a way to try and find one possible solution or several possible solutions for the sustainability problem that Peter alluded to. And for the case of TARE, which is what we started with, we've been able to solve that by getting the global user community to support us by subscriptions. And so that's universities, individuals, lab groups, some companies to contribute to funding the effort of growing and maintaining the resource. And then after that, we transitioned into trying to help other resources who were in similar straits find a solution as well. And for some of them, that has been subscription. So Biopsych is one and RepBase is another. For another resource called Morphobank, which deals with morphological data matrices used for phylogenetic tree building, the solution has been through membership, where institutions join and their fees help provide open access for everybody. And then there's another resource called Cypress, which is a phylogenetic tree building compute resource where people buy blocks of hours. And then that type of subscription helps support their operations. So my path has been from kind of curation at a single resource to being involved in many different resources of different types of biology. And then I ended up training in. And so from reading papers and working with communities and ontologies and curation standards, I've now kind of been able to learn through this transition to Phoenix skills in HR. Who knew things about insurance? Now I do. Who knew things about kind of liability and things that you and contract negotiations. And then now I'm also curating, in addition to the little bit of curation I still do in the plant biology world, I'm also curating in the world of morphological data matrices, which is a whole different area of biology and paleontology. But the wonderful news, I guess, to share with everybody is that all those skills of being detail-oriented and being able to dig into a paper and learning how to interact with databases were totally transferable to this new domain. And so while it may have seemed for me a little intimidating in the beginning, it was like, okay, Terra is a database, Morphobank is a database. I know how to do SQL on this one. I can look at that schema and figure out what to do. So that's my maybe 3 or 4 minute introduction to how I got to this panel. Thank you.
All right, great. So I'm sure we can get into some of these issues later. I mean, we don't have too much time, but let's try to discuss some of these ideas later. So the next one on my screen is Pankaj. Can you please introduce yourself and the resources you're curating and how you ended up in biocuration?
Thank you, Pierre. This is Pankaj Jaiswal from Oregon State University. I'm a full professor there now, and I started with my background in biochemistry, as usual for all the other PhDs that we see in the biology stream. And my emphasis area was mainly on plant molecular biology and plant biology. But then later on, as we were started sequencing some of the molecules, then more of the bioinformatics analyses, curation, and genome biology started picking up. And now my lab does both wet lab and a dry lab projects on all sorts of things. And 2 major projects are working on curation of gene functions, phenotypes, pathways, and gene expression data sets for the Gramine database, and especially with the plant Reactor version, which is built on the same principles, and we curate all the information with the human Reactor group. But we take the plant slice, so some molecules are common, but the pathways and reactions, sometimes the reactions are also common. So we do co-curation, and then we take the plant slice and produce that database. So it's a whole team effort that we have. And then another one is as newer plant genomes, about 200 or so plant genomes are getting sequenced. A lot of phenotyping information is coming out, and a lot of genome-wide association studies coming out. I think there was a lot of data new data that's coming in. So there's a need for capturing the standardized information of those information. So I started the projects on ontology development for plant biology, known as Plantiome. And these include the developing of ontologies for anatomy, growth stages, traits and phenotypes, experimental conditions, and sometimes they are generic or species agnostic, but many times they have to be provided in a species or clade specific manner to address the community needs. So we have crop ontologies. And then we are building on the plant stress ontologies in terms of curation aspects. In terms of what you asked me, how did I come into the curation thing? I would say that without knowing what curation is, actually, we all started in our masters or in PhDs, and we started reading papers or capturing text and information and coursework. And our goal was to learn and synthesize the information to address a biological question. So that's how it all started. And then as this is an evolving process, as you grow into your experience, you talk to more peers and networks and learn, and as the research is evolving as well. So I think whole goal was, what is the easiest way and the best way of synthesizing, but also of easy interpretation, inferences, searches, and how to access the information. And I think that got into the curation aspects of my, as I entered the Grameen database and the Ontology's database, as I was seeing. And I've learned a lot from our model organism databases and the gene ontology curator folks as well. So the emphasis was always on what do we know and where the deficiencies are and opportunities. So that's a critical thing, that curation is one aspect, but curators, as you can imagine, they read hundreds and thousands of papers, many more than anybody else, right? And they know ins and outs of how to synthesize information and what is the actual information that's lacking in a certain gene family or a gene set or in a genome. And that prompted me to, gave me the motivation, can I have my own lab and go test some of the stuff as well, and motivate my students and postdocs and researchers on training and education so that they can do some work in a standardized way and learn that whole process. So key is also on training, education, and supporting our new generation of students and researchers so that we can build on a great work and the best practices that the biocuration community has developed in general. So that's my spiel for that. Thanks.
All right, thank you. So I'm sure we can get also into some of these issues. So Nicola, you are, involved in a Pan-African project, and I also saw that you co-authored a paper on how to organize a webinar, which I should have read probably before that. So why didn't you tell us your story about how you got into biocuration and what are you doing in terms of databases and so on?
Sure. So I am not actually curating any databases right now, but I'll come back to what we're doing now. So I did my PhD at the University of Cape Town in wet lab work, actually. And then towards the end, there was quite a lot of bioinformatics. So I was really interested in going into bioinformatics. And I saw this position open for a Swiss-Prot database curator at the EBI, European Bioinformatics Institute, which most of you know. And I went there in early '99, and I learned how to be a Swiss-Prot curator, but that didn't last terribly long. I was there Swiss-Prot curator for about 6 months, and then the Interpro project started. And they needed some people to work on that. So myself and one of the other brand new curators who started the same day that I did at EBI, we were moved on to Interpro, and we basically spent months just integrating Interpro entries, curating them. And from there, I kind of ended up leading the project. So I was just the project coordinator and then an official team leader at EBI leading the Interpro project. And then under the umbrella of the project was also the Gene Ontology Annotation project at EBI, who was mostly doing human annotation at the time. And so while I was starting to move away from curation, we obviously had to build these curation teams because Interpro had, the development team and the curation team. But, as a biologist, I was always pretty interested in the curation side. And then while I was at EBI, I've also always been interested in sort of functional classification. And before Go came about, I know we're talking many, many years ago, there was the Drosophila paper, genome paper, and there were, was a functional classification there. So I actually developed my own functional classification scheme for Interpro entries so that you could use Interpro to functionally classify proteins that you're annotating. And then when Go came about, we then started mapping to Go instead, the Interpro entries. But this original functional classification scheme was actually used in the human Nature paper, the human genome. And so I've always been interested in that. And then so I was really into Pro, and then I really wanted to come back to South Africa and move back into research. So I came back here and very quickly sort of moved, although I've got a huge research group going on in various areas of diseases, I in parallel moved into the stayed a bit more on the services side and building resources because I really actually enjoyed that. and became involved in this big Pan-African project. So we already had an informal African bioinformatics network before H Africa came about. And then when H Africa came, I put in the grant, and we now have this funded Pan-African bioinformatics network that builds capacity for genomics research in Africa. And so while a lot of our activities are about supporting bioinformatics, in terms of the curation activities, I've kind of switched to the user side of curation, but also, implementing use cases. So for example, we identified a need for, one of the projects we work on is sickle cell disease. And we identified a need for sickle cell disease ontology. So we got the entire, the global sickle cell community together to build this ontology together. We subsequently built one for hearing impairment. So there's a hearing impairment ontology. But really in H3A Bionet and our genomics activities, we do quite a lot of curation in terms of building and implementing standards for phenotype data and having those curated in a good way, because we've got multiple different cohorts, research cohorts working on different diseases. And then we map those to ontologies, including the cohort metadata, which is mapped to the new Gecko ontology. And then we also have a resource called we're building a resource called the Genomic Medicine Portal, and there we're extracting data from multiple different databases, which many of you curate, and then trying to curate these for African relevance. So those that are particularly relevant to African diseases, have SNPs from African populations. So we sift quite a lot through the literature, through database metadata to try and curate these resources. So the curation work that my team is doing at the moment is very mixed. It's not one database that we curate. We use curation expertise in multiple different things. We curate training materials, for example, by mapping to bioschemas. So all aspects of the genomics sort of environment and ecosystem, we use curation in those.
Okay, thank you. So we have heard multiple times from you and from other people, that curation is really a multidisciplinary and actually an international process. So the question is also, how can we get people involved, since we are talking about careers? And some of you, including Sushma before, you talked about like getting students involved. So I think a good way to get into curation is to volunteer for one of these existing resources, although that may be a bit of a tricky business because you need some skills and some experience. But so one of the questions I had for you guys was, do you have any recommendations how people can get involved by volunteering for a database or for helping out? Like we heard about students curating and things like this. And I think that's probably a really good opportunity for people to get their hands wet, and see how the whole operation works and if they like the whole operation. So let me ask you, Nicola, first, because you are involved in this Pan-African project. Do you have people all over Africa helping with data curation or helping with data resources, and how you go about that?
We do, yes. So even though it's a Pan-African, it's a bioinformatics network, there are quite a few people in there who are very junior and don't really have the technical computational skills. But they do have the biological background. So we do this through hackathons, data jamborees, but sometimes, we have a set of data that we need to curate, and then we call for volunteers, and then we just set up the tools, and we usually just work in very simple environments like Google Docs, Google Spreadsheets. We sort of provide a bit of training on how to do this, and then the curators do this. And then when they come unstuck or when they're not moving very fast, then we actually have an online meeting, an online curation session. So the curators all come online. They don't necessarily have to talk to each other, but they're just all present. And then you can see all the curation happening, and they can ask questions where they need to. So we've had that, and then we've had other events where we just need to annotate a few things, and then we just open it completely wide and say, we've got some terms or something, we need them annotated, and we need people to read papers and report on these. And then we get volunteers literally from across the continent to come in and contribute. And there's some where we try to, got a genomics catalog, for example, where you can actually put your name and your ORCID ID, so that, if any annotation is done, even if it's, a small bit of curation for a particular project, that you are recognized for that, and that people, they put it on their CVs. So we do try to have, at the moment it's mostly online, but we've had a face-to-face hackathons and data jamborees where the curators all come together and sit in a room and do the curation. The ontology, the sickle cell disease ontology development, we brought in experts, clinicians, really busy people from around the world, put them in a room, and they just had such an amazing time giving their expertise in building this ontology, in real time. So it's quite, that we funded their travel and we gave them nice dinners, but we don't always have money to pay people to do a bit of curation, but they feel that if they kind of have some sort of recognition on their CVs, either their ORCID ID is attached to some sort of curation effort, then it benefits them in the long term.
Yeah. Okay. Well,
Nicola, Nicola, may I follow up on the ORCID recognition? So you have the ORCID ID displayed maybe on one of the databases, but is there also a connection to the ORCID record itself where then it's like shows up as one of their works?
So only if there's a DOI, I guess, associated with the record or the database or whatever it is that they curated. So it's usually more for their CVs. And then if people search that ORCID ID, then they can identify that that is the same person.
Yeah, that's a really important thing, how to get recognition.
Thank you.
All these efforts. I totally agree. And so one of the things I'm doing, for example, is I teach students how to edit Wikipedia pages so they get at least a bit of experience, translating a paper into like an annotated piece of information, although that's not structured information as in a database, which brings me to Pankaj, because I was just looking up the number of open reading frames on the Wikipedia page for Arabidopsis, and I realized that, some papers, including Chris Towns' paper, say there's 27,000 genes, and UniProt says there are 39,000 genes. So how do we reconcile these things, and how can you get like students or volunteers involved in your multiple operations, Pankaj?
Yeah, so that's a great question, and I think we have seen those kinds of information coming up, but I say it's a wider conversation needs to have within the community, as well as at the data archive places. So for example, my Grameen database project works directly with the Plants Ensemble group, and all the source for peptides, for example, in the ensemble group, Plants Ensemble, is the UniProt. They do carry out all the baseline annotation for that. So ideally, every gene, protein coding gene, for example, in Arabidopsis or in rice or maize, whichever version we are looking at, is expected to have at least one equivalent entry in the UniProt and back and forth the sharing of the information. But there are additional legacy data sets are also there, and the newer versions of the genomes, 5 to 10% of the genes get dropped or picked up a newer one. So the numbers may vary, but I think their proteome set should be consistent with what's the newest version of the genome. And I think there's a lot of information that's to be discussed. We have tried to enforce it, but oftentimes I think it's also a conversation with the publishing houses, where the genome is published, for example, the raw sequences are deposited in the archives, the genome assembly may be in the GenBank, but the actual gene annotation or gene calls, they are not in the GenBank. So unless those are there, then, so I think it's a data standard, data archive conversation, and a public education and training on that side, that if we can come together on some kind of a consensus to build on it.
Yeah, I mean, as I said, I found the reference proteome in UniProt having a completely different gene number than you see in some papers. So, so that brings up the question of how you go about these issues if you see that as a user or as a volunteer. In fact, Shaunak Bakshi is asking in the Q&A whether there are portals, to know about volunteering opportunities, and I would suggest, people or students who are interested in that, they should just directly contact the databases they are interested in or the resource. If you're interested in plants, email Pankaj, about Grameen or whatever database he's or the TAIR, of Tanya. I don't know if Tanya is still involved in Arabidopsis,
Absolutely.
Yeah, and
it's still our main project.
Yeah, which we can use as a transition directly to your topic, because many of these databases are not funded. That's a very common issue on databases. I mean, there was this report in Science a while ago which said that even like model organism databases have trouble getting funded, you know, and so you developed some mechanism to solve exactly this problem. Can you tell us a little bit of how you went about that?
Well, it was mostly desperation, right? We wanted to keep serving the resource to the community. And so we were like, okay, how are we going to do this? And so, you know, various solutions from, we're going to find some kind of angel investor. Let's find some, you know, big name philanthropist who thinks that this is a great idea and will give us money, and kind of evaluating, which we didn't find, right? But then just kind of thinking, how can we support it? And one idea was subscriptions. And then it was the question of, are people going to be willing to pay, right? If they've been used to accessing TERR for 10 years, 12 years for free, are they going to be willing to pay? So our experiment was, okay, let's see. We'll put our jobs, basically, we'll put our money or we'll put our hopes on the community and say that this is a resource that's been very useful for you. Are you willing to help keep it going? And the benefit of the TERR community is that it was very large. So not every resource that's out there that's running out of or is running short of funding will have the same type of community that TERR has. And so for us, subscription was a workable solution, right? So we were able to spread out the costs.
Yeah, let me interrupt for 1 second. So TERR is specifically about Arabidopsis, which, how large is that community? How many people or labs are involved in?
About 10,000 labs.
Oh, wow, that's a huge
all over
number.
The world.
But it's still a model organism and a model organism database. So,
absolutely.
But it's not the thing. I guess if you're working on a crop plant or something which is commercially interesting, proteins or human disease, it's much easier to get funding. But Arabidopsis is, I guess, mostly an academic thing. So how did the academic community respond to your efforts to commercialize or base it on a subscription basis?
Right. So initially skeptical, right? It's like, why are you, why can't we keep getting this for free? And the harsh reality is that the government funding agencies were like ready to set us free, so to speak. Go ahead, you're ready to fly. And so there was some reluctance on behalf of some academic institutions to support, and we understand that. And also the idea of like why we started Phoenix as a nonprofit was so that it was clear to the community that we're not in it to make a buck. We're in it to help sustain and grow the resource. And so when we were doing kind of the initial pricing evaluation, we were really trying to keep it at what do we need to sustain our budget so that we can have curators adding to it. And kind of over time, the community, at least our community, has come to accept that, yes, this is an acceptable cost, like buying restriction enzymes is, like paying for a journal publication or paying for a journal subscription. And that's how we've kind of gotten into the academic libraries supporting TERR, is by them recognizing that this is a resource like a journal subscription or like another type of digital resource. And over time, now Phoenix has been around for almost 8 years now, 8 years, because we started in 2013. That awareness and that support and acceptance, kind of beyond acceptance, but like actual active support for this model in TERR has grown. Pankaj.
So can you provide a few numbers, like how many subscribers you have, or maybe even disclose some of the fees you charge for using a database, so our listeners get an idea of what we are talking about here more specifically?
Right. So if we look at everything, all our pricing schema is public on the website, so phoenixbioinformatics.org, if you want to dig a little bit deeper. But if you're subscribing as an institution, it's based on usage, past usage. So the more you use, the more you pay. And then once you've established a usage, you can use it as much as you want. So it's something like $4,000, $4,500, I think, for the highest usage a year. And that covers everybody at the university. And we check by IP range down to, I think, something like over, like the lowest tier or the cheapest tier is less than $1,000 a year. And then if the university can't support that, There are individual subscriptions that are available for, I think it's now just over $100 for a year of access. And we have probably in the 200 or so universities that are subscribers. And I don't know what the numbers now for the individual subscribers, but it's probably in the 100, 200 as well.
Okay, that's not bad. So given that there are tens of, I don't know, if there are thousands of databases worldwide, and I don't know how many of those are really funded right now, but it may be an interesting option for a lot of these. I mean, how many databases have actually contacted you or shown interest in your subscription model?
I would say that we have had conversations with in the tens of databases, so probably 30, 40. And that's just kind of exploring. It's like, what are you guys doing? We want to share our knowledge. Is this interesting? And that's part of our mission too, is just to share our experience, and whether or not we end up partnering with that database. If we do, great, but sometimes our advice is this may, like, subscription may not be the best solution for you, but here's what we learned. Maybe you can take from that as well.
Yeah. So for those of you who are listening, that's an important issue, because if you want to work as a curator, either you have to do it as an unpaid volunteer, or as someone who is paid for something else and just doing it on the side, or really get a job as a curator. So Pankaj, how many people can you pay for your resources to work as a curator, in one of your projects?
Well, if I added that, hardly 2 to 3, depending on the number of resources and the amount of funding in a given year that is available. And we have, let's say, 100 plant genomes. So it's absolutely that it's minuscule in that sense. So I think that's why the question that you initially raised is on the how can community contribute to the curation aspects. So obviously this seems like a volunteer, but I think what we need is the community's help. And the best way to start is that if you find that there is you are interested in and reading papers, and you find that certain information is missing in a database, the best thing is to send a feedback and get that process rolling. If you publish a new paper and provide that information, reach out to them, because the curators are already overwhelmed with a lot of information. So the idea is can you reach out directly to them, and they will be more than happy to come and prioritize, because this person contacted you personally, and they will get on to that thing. Work with the databases to make sure that your data is prepared in a proper manner and for completeness and efficiency, well before you are actually ready to publish. They can keep it behind the firewalls and not make it public, your data, but they can still process your data very efficiently. And maybe the authors can actually get access to that early analysis, that more in-depth analysis that these databases like Plants Ensemble, which would actually, if somebody runs a genome analysis, they would do the analysis of gene families against their own closely related 2 or 3 or 4 or 5, 10 species. They would give you the data for 100 species. Can you do a bigger analysis on it? So there are multiple choices. On the other hand, what we have done as a community aspects is we have brought the biocuration by co-authors at upstream level, where when the manuscripts are accepted, majority of the resources, what they do is they process and do the natural language processing and machine learning after the paper is published. We went ahead and did the upstream value. And we said, okay, as soon as the manuscript is accepted with all its revisions done, and it's in the final proofing stage, at that time, we run an NLP or machine learning to find the entities like genes and phenotypes and ontology terms in the manuscript. And and as the author is approving its proof, it will trigger a process saying, do you approve the findings that we have from this paper? If not, can you go ahead and edit or delete? And that is, some authors are overwhelmingly engaged. Some are just do the minimal. So we give them both options, and whatever they curate, that data gets automatically annotated on the published paper online, which is much easier now in a standard format to extract that information as well. So that's where we have engaged close to around 900 community members. I don't know how many were unique, but 900 papers were annotated for just 2 journals in one single year by those authors. And that process continues to evolve. So I think engagement is necessary, but don't trust that resources have everything that you expect them to do. That should be the way.
That is a really important issue, which we should turn into a separate community curation panel discussion separately.
But if I may add one more thing on the commercializations aspect versus subscription model versus a non-subscription model, or go completely public open source model, I think for some databases and resources that are very much contained within themselves, then it's much easier to protect the IP and protect the database and the resources. But for example, Grameen and Plantium, where hundreds and thousands of users are using, much of the data is integrated from external sources and some value added through internally. And whatever we serve, for example, Grameen's genome portal is co-produced and mirrored from Plant's ensemble and Grameen itself. There are 2 places, the raw data and all the sample data is in UniProt as well. So it's very hard to protect the subscription. If they don't get it at Grameen, if let's say Grameen goes paid, then the other resource is already free and and data is available in bulk. So you can't protect all those kinds of things. So there are challenges, but we need to be very careful on where we want to go into the subscription versus distributed data annotation and data sharing model that we're looking at. Thanks.
All right, thank you. So there's one question from the chat here, which is about, do we have an example of an open access publication where this kind of annotation is available? I'm not exactly sure what is meant here, but I mean, one of the problems is we have to release stuff open access as public databases, but at the same time, if we charge for access, we cannot release everything. So I guess maybe Tanya can say a sentence or 2 about, like, what is her experience with, how much of a database should be public and how much can be commercialized or put behind a paywall, because we have to find some compromise here, I guess, to make people at least be able to use the database to some extent, but not release everything, right?
Okay, I found the unmute button. So to answer the question about how much to make public and not, our solution for TER has been to make our curated data available for public reuse under CC BY after it's been in the database for a year.
So your microphone is a bit difficult to understand right now.
Is this better? Yes, great. So the TER data is made publicly available, the curated data, after it's been in the database for a year. And so the subscription incentive is you get the data that's freshest and most recently curated. But if you're, if you're willing to take year-old data, you can take it then. But also we provide free access to a limited number of pages every month. So if you're a casual user of the database, you don't have to subscribe. You can just come in, look at your gene of interest, run your one analysis, and then you're done. But if you come back using it more often, then it's like, please support our resources. I wanted to address one of the questions that's in the chat, and I think the other people on the panel would also be interested in commenting on, which is, let me just read it now. Given the sustainability issues, how do you see careers in biocuration evolving? I can tell you that from my experience, right? I started in curating in biology, in plant biology, and then it's become a little more broad. But as part of the Phoenix transition, or of Phoenix's involvement with other databases, I've become exposed to curation efforts in other areas of science. So curation efforts in paleontology, in other geosciences, in chemistry. And I can tell you that from the conferences I've attended, the problems that they're facing are the same. The types of attention to detail that are necessary for the curation of data are the same. And that the skills that one learns in biocuration even if one doesn't stay in the field of biology, are applicable to these other fields as well. So evolving could be you move from biocuration to curation in another science. It could be that you move from data organization in science to organizing different types of data. So I think, sustainability you can look at from various different aspects, sustainability of the resource that you're curating for, but also sustainability of the job that you yourself are doing, right? So that's my offer. I know Nicola chimed in too about wanting to add to that.
Yeah, let's have Nicola make a few comments on this issue, especially from her perspective.
So, I mean, there's two aspects. One is sustainability of these big resources and the curation aspect of those resources. And, Tanya's covered that quite a lot and has one of the solutions. I think people are recognizing the importance of these databases and the curation effort. And so I think that's why organizations like the Global Biodata Coalition has come together to think about how to fund these long term. But in terms of other sort of career paths for biocurators, I see the need for biocurators and data stewards just increasing because everyone is talking about fair. Now you can't just make data fair. You actually need people to do that. And it's actually people with curation skills that are the best place to do that. So in various projects that I've been working in, the data sets are getting bigger. Data is becoming, the new oil, and it's absolutely useless unless it's curated and you have that value add on top of that data. So I see that as the data sets get bigger, the need for curators in a different sense, not curating databases, but curating data sets increases. Now, obviously the funding is a problem because most people who are generating these data sets don't actually put a line item in for that curation. But in the discussions I've had with various funders, they're going to start insisting on a budget line for making your data fair, which means curators, which means data stewards. So I think, if you're willing to step away from those traditional curation of those databases and think about curation and helping people curate their data sets, I think there's going to be a huge future need for that. And there will be a push for this to be in the budget, so that there is sustainability for such careers.
I also would like to emphasize the fact that, all the scientists or, biomedical scientists in particular, we are all supposed to be biocurators because we all read papers or a lot of papers, and we should also we also use databases, which we also see that these databases are not perfect. They have mistakes, and a lot of students I have, they just assume that the database is perfect, it's complete, has all the data is flawless, and so they have to realize, everybody can actually contribute. And some of you guys have mentioned before, everybody who uses a database can contribute, email the database itself. There's usually a contact link in database entries, which you should use. And that's your first step into biocuration. For the database I maintain, I get like a couple of emails every day. I have no idea how many emails the big databases get. You guys tell me, how much interaction you have with volunteer curators who just inform you about gaps or updates in your data sets. Can you comment on that?
We get maybe 2 or 3 a week.
And so if you
it varies,
yeah,
from you missed a publication or you missed a symbol or here, please do this.
So one question is, of course, depending on the database, if you do not get enough feedback or not enough as you may want, how can we encourage people to contribute more or to volunteer more as database, volunteer curators, let's say? How can we get the community involved, besides just the accidental people stumbling upon a problem or a mistake in a database?
I think incentives. So the incentive, if you provide training, then that helps their CV. If there's some sort of recognition, then that helps their CV. Just joining a community. Some people are reasonably isolated where they are and being involved in that. But then, if they do volunteer, then help to build that community. So help them to feel a part of that, that the contribution was important. So I think incentives are important because people, not that many people just have a lot of spare time to do this. But if, they receive training and it opens up other doors, maybe they get to meet people or other curators, I think that is all incentive to contribute.
Yeah, Sushma just said that, it's also important that databases work with journals. Grameen works with different plant journals, apparently. So I think that's a really important thing. So if you're involved either in a data source or in a journal as an editor or as an author or as a publisher, or whatever. So it's really important that these data sources talk to the journals which we are actually supposed to curate. So I think that's a really important thing to do for all of us. Let me see what else we have now. So are we going over the hour, or we just do the full hour as we were planning for?
I think it's probably good to start wrapping up soon and maybe have some final thoughts from the panelists before we close.
Yes, just let us know if you have any particular recommendations for students or other people who are interested in becoming more involved in biocuration. What would be your recommendation to get into this field, or how to start, even finding a job in this area? Let's start with Nicola again, because she hasn't talked that much, and you have all these interactions across the continent of Africa. Do you have any recommendation what people can do in Africa and beyond for getting involved?
I think draw on your area of expertise. So there's always room to learn new things, but what you have to offer is your area of expertise. And in the first instance, if you can't find a job in curation, volunteer for some of these efforts, because if I see somebody's doing a great job, it's something that's going to push me to try and find a position for them. So I think do as much training as you can. There are a number of courses available now, and think about where, if there isn't a big database in your area that you can curate for, or maybe some online efforts, then think about other avenues like, as I was saying, in our cases, they're big data sets that need curation. So, on an individual cohort level or an individual research level. The other way to think about, where to go in curation is search for job adverts and look at the job description and think, am I qualified to do that job? And if not, how would I need to build my competencies to be able to apply for a job like that? And there are courses that you can do. So even if you're not totally sure about what your curation skills are now and what value they are to other people, just by following those jobs. So if Grameen's got a job opening, or if EBI's got a job opening, look at the job description. Think about whether you fit that job description or how you can build your skills to do that. But as I say, volunteering, putting yourself out there. If I see somebody who I've worked with, even if it's in a volunteer capacity, or, I've seen their face, I've seen some of their work, I'm far more inclined to take them on next time for, when I have something available. So I think just grab every opportunity that you can, put yourself out there. And build the skills. There's a lot of different skills that are required because there are a lot of different types of biocuration. And I think if you start thinking about what direction you want to go and find job descriptions and build your skills in that area, I think you can make yourself a lot more attractive in this field. And I think it's an amazing field because they completely, as you all know, completely underappreciated, but I think it's one of the most important jobs in terms of adding value to data.
Yeah. All right, thank you. So Pankaj, final comments?
Yeah, I think I agree with Nicola. I think what I would say that biocuration is important, and the good quality data is the gold mine for doing everything, including your AI and machine learning. You cannot compromise on the biocuration activity. But however, the newer students and researchers that are coming in, whatever information you curate, but you remember there is a lot of open data available to you. So if you can add your data analytics skill on data mining and how to navigate through that clutter of data and find the real good questions and biological paths and knowledge inside that would be the key, and that will set you up for a new job, for example, in data analytics or things you may not find, or maybe very less number of biocuration jobs, but data analytics needs it, and whoever knows the biology and data analytics and curation, they are the key to the future generation. But on the other hand, if I may add, is that everybody, all the agencies and all the projects are supporting students and training education for postdocs and things. then everybody's moving on to the newer and bigger projects in developing large scale data sets and machine learning. But somewhere in the middle, we forget that there's a lot of biocurators that are available for which there is absolutely no support if the funding goes away, and that quality of the data and the resource get compromised. So I think we needed a concerted effort from the community to alert the proposers and agencies to tell that, hey, biocuration community is the key and we need support. Otherwise, we don't find the funding mechanisms to support to these people, then all the downstream and upstream processes get compromised in many ways. And we keep saying that there is a lot of data and we should go on to machine learning, but you can't actually, the good quality data is minuscule. There's not enough data points to carry out the AI in biology as of now. There's no good quality sanitized data. So we need biocuration in that sense. Thanks.
Yeah, thank you. So I completely agree, and we have a whole range of activities going from volunteer to full-time curator, and from just technical curator to like bioinformatics analyst. So there's all these, things in between. Now, Tanya, you have any last final comments on this?
Just to follow up on the volunteering part, is the volunteering is an opportunity for, if somebody is exploring biocuration, is to figure out if you like it, because there's a certain personality, and I know a lot of you here have that personality, which derives extreme satisfaction from organizing stuff or finding that one little hole that's been there forever, and you're going to fill it. And that feeling is amazing because you're filling it for the community. On the other hand, other people may look at that type of job and be like, this is drudgery. And so if you find through volunteering that this brings you joy, then you know that biocuration is for you. So that's an opportunity to explore that without starting a job and then realizing, oh, that was not right. That's my final.
Peter, you are on a
Sorry,
mute.
Peter, you're muted.
Yeah, Peter, you're muted.
I was just muted. Sorry about that. Yeah. So I think we can conclude this discussion here. I just wanted to point out that you can save the chat by the little ellipse in the bottom right of your screen if you want to keep the links to various papers some of the participants have posted and some of the other comments. So we can leave this open, this window, for like another minute or so you can save your chat. Sue, do you want to make any final comments or announcements?
Yeah, so I would just like to thank all of the panelists and speakers for a really wonderful session today. And remind everybody that the next session is in August. So please submit your abstracts. And really, again, thank everyone for participating today and wish you all a lovely day.
All right. Thank you so much.
Thank you.
Bye.
Thanks, everybody.
Thanks, everyone. Good to see you.
Thank you. Bye-bye.
The session will be recorded and will be posted on the ISB YouTube.
Yeah.
So if you want to come back to anything later, it will be there for you.
Thank you.
Sounds good. Have a good day, everyone. Bye-bye.
Bye.